First of all, next time we meet, next Tuesday, there's a modified class schedule. So we will be starting at a different time. So keep that in mind. What that time is, I don't remember off the top of my head. I have posted it on the homework slide in this slideshow. What the time is, I believe we start at 9:30. So I think we start a little earlier next Tuesday. But we also end a little earlier. Right, because of the election day, there's a modified schedule. So that if you want to vote in any elections, you have the time and ability to do so. Okay, so keep that in mind so that you don't show up late to class next Tuesday. We are starting at a different time. All right, the next announcement I had, so the scientific literacy assignment is due on Tuesday. Right? And I noticed that the link to the library has changed a little bit. The link still works, but the how it looks is different, right? So the instructions within the assignment are slightly different than what I have typed initially, right? Because I redid the website in August, and I did not catch this change. So what I'm going to do is go through and slightly update those instructions so you can navigate to the library's database, and I will email you out those updated instructions to help you, right? So you might be able to do this without those updated instructions. You might be able to figure it out, but in case you can't or you just don't want to, keep an eye out for that email. All right. I think that's all my announcements.
Oh no, I need double these one.
Any questions? All right, we're actually going to skip this slide because this class got to this slide last Tuesday. All right. And we're going to go right on into DNA replication, which is our next topic here. So we've talked about DNA and how it's structured and how we get chromosomes. Now we're going to talk about how these chromosomes can replicate, right, in order for a cell to divide, right? So when a cell divides, it needs to replicate its DNA so that when it ends up being two cells, each of those cells has a copy of the DNA that was originally in the parent cell. Right? So when a cell divides, it needs to replicate its DNA. Right? So most of the time, our cells are not just randomly replicating our DNA. It's only when that cell is ready to divide. Now, it might divide via mitosis or meiosis, and we'll talk about those next week. Right? But either type of cell division, we need to replicate our DNA. All right, not too much on this slide here. So when we talk about DNA replication, there's a couple of terms that I will keep saying, right? I will talk about the parent strand and the daughter strand, right? So what am I talking about when I say those terms? Well, if we look at this image on the screen, right, we have one double helix, right? That is the original double helix that we're going to use in replication. This would be the parent strand. They contain two strands. Each of them are the parent strands, right? When replication has occurred, and now we end up with two sets of DNA, each set is going to have a strand that has been synthesized, that has been replicated. In this picture, it is orange, right? This will be the daughter strand, right? Now, one of the homework assignments that is due next Wednesday, actually, I gave you an extra day because I don't want everything to be due on the 4th, is the history of DNA, right? And one of the videos I'm going to have you watch is the experiment that led us to conclude that this is how DNA replicates. How we go from one parent right, to two double helixes that has one parent strand and one daughter strand together. This is actually known as the semi-conservative method of DNA replication. There were originally 3 theories, and we landed on this one as the one that is accurate. And you'll learn about that in the assignment, right, in that short video that you will watch, and you'll learn about that experiment that led us to that conclusion. All right, so we have DNA replication. Where might this occur? Where does this occur? In the nucleus, yes. We know DNA stays in the nucleus. It cannot leave the nucleus. So this whole process is going to take place in the nucleus of our cells. Right? So DNA replication takes place in the nucleus. All right. So now we know what parent strands are and what daughter strands are. Let's do a very broad overview of what we will be learning about, the process we will be learning about, which is DNA replication. Right? That's where we have 2 parent strands. And in this image, the parent strand is the one that starts as a double helix, and it's color-coded as red in this image. And the daughter strands are the ones that are made off of the parent strands. So the parent strands act as a template, and then the daughter strands are made according to that template, and that is blue in this image here. When we're going through DNA replication, right, we're still going to abide by complementary base pairing. Right? So A's are still going to hydrogen bond with T's. C's are still going to have hydrogen bonds with G's. Right? We're still following the ATGC rule. And at the end of this whole process, we're going to end up with 2 molecules of DNA that should be identical. Right? So we're starting with our parent strand. That parent strand is going to divide. 1/2 of the parent strand will be part of one of the new DNA molecules, and the other half will be part of that second new DNA molecule. And those molecules that we have created should be identical. All right. So how does this happen? We are going to start at the beginning. Right, so DNA replication is going to start by forming what's called a replication bubble. Right? We're going to start at the origin of replication, where the DNA is going to unwind a little bit and form this bubble of unwound DNA. Right, so it's this particular sequence in DNA, right, called the origin of replication. At that sequence, the strands will start to unwind to the point where we have what looks like a bubble in the double helix, right, of 2 strands of DNA, where the hydrogen bonds keeping them together have been broken, and now we have this bubble. Now, on either end of this bubble, we have what's called a replication fork. Right? And at the replication fork, this is where DNA replication occurs. Right? So we're unwinding a bit of DNA, creating this bubble, and at either end of this bubble, right, is the replication fork, and that is where replication is going to occur. Right? So we have to start somewhere. This is where we're going to start at this replication bubble. Now, because it has 2 replication forks, this means that DNA replication is happening in opposite directions at the same time. Right? It's going in one direction and simultaneously moving in the opposite direction, replicating at the same time. Now, when we talk about DNA replication, we're only going to talk about 1/2 or 1 replication fork at a time. Right? But I want you to understand that this happens at both ends simultaneously. All right. as we talk about DNA replication, there's a lot of proteins involved, a lot of enzymes involved. And that's how we're going to walk through this process, by talking about the protein and what its function is and how it plays a role in DNA replication. So the first protein we're going to talk about is DNA helicase. So DNA helicase will come into play at the replication fork, right? And it's going to continue to unwind our double helix. Right? It's going to continue that unwinding. So what it does is it actually breaks those hydrogen bonds between the base pairs of the two opposite strands. Right? So we're breaking apart these this double strand into a single strand. We're breaking apart those hydrogen bonds. Right? So it kind of acts as a zipper. Right? So if you were zipping something up, that tag, that zipper that you pull, you can think of that as the helicase. It's going to unzip the double strand. All right, so helicase is doing the unzipping, but once it's unzipped, you have to make sure it doesn't go back together into a helicase, right? Because if we just left it alone, these two strands that have been unzipped are going to want to reconnect and reform that double helix. They're not going to want to stay apart by themselves. Which is where our second set of proteins come into play. Right? This is actually a grouping of proteins, and a grouping that we call single-strand binding proteins. So in order to keep those strands separate, keep them unzipped, these single-strand binding proteins are going to bind to those unzipped strands to prevent them from coming back together into a double helix, right? To keep that part of our DNA open so that we can proceed with DNA replication. So it kind of stabilizes it. Right? Just like if you were to have a zipper, right, and you unzipped it, and then you put a clamp or something on part of the unzipped part. You were to try to zip it back up, it's not going to work, right? Because there's going to be a blockage in the way. Right, that's kind of how these proteins work. All right, so helicase has unzipped our DNA. Our single-stranded binding proteins have kept the DNA open. We have one more protein, right, helping to unzip and unwind our DNA. And that protein is actually called DNA topoisomerase. It's a nice fun name to say here. Right, so as we're unzipping our DNA, right, helicase is doing the unzipping, our single-stranded binding proteins are keeping it open. Around the double helix itself is this DNA topoisomerase. So on the opposite side of helicase is this topoisomerase. And what this enzyme does, right, is it prevents supercoiling. All right, so if you've ever had a piece of string that has like 3 strands woven together, and you try to unweave those strands, or you had a tangle of cords, maybe it's your like headphone cords. I know everybody's cordless these days. Everyone uses Bluetooth, but cords, your laptop cord, it's all in a jumble. It's in a mess, and you're trying to untangle it. And if you pull 2 ends, right, at some point, it's not going to pull apart anymore. It's just going to become a knotted mess. Right? So if we were to take apart like a strands of a string, start to pull them apart, there's going to become a point where it's become so super coiled behind where it's breaking apart that it's going to become like a knotted mess. It's not going to want to pull apart anymore. So topoisomerase prevents that knotted mess from happening, prevents that super coiling, right? So if you go back to your dorms, find a piece of string, or try to untangle some cords and you're pulling 2 ends and all of a sudden you just can't pull anymore, it becomes knotted, right? That is what topoisomerase is trying to prevent, because that's super coiling. Right, normally I would bring in a piece of string and demonstrate that. I forgot it in my office. So if you want the demonstration, come to office hours this afternoon and I can demonstrate it for you. Right? But it's preventing that super coiling. All right. Let's take a break, review the proteins we just learned. Tell me, what has DNA done so far, and what are the proteins involved?
It's like zipper magnet. It's like a mochi. Zipper magnet. Just sorry. Like a tablet. It's but it's like a
What?
Oh, got it.
So it's called also isomerase.
It's actually I also want to
I love it. I love it. Fantastic.
which classes? That's so cool.
I have a mini like sticker printer.
How do you use it?
I'm gonna try to I can like photo.
Wait, that's so cool.
Yeah, I got it.
Wait, you do?
Yeah, I got it. All here. I don't know, I forgot I have a question. You can make like stickers and many stickers on this. There's I really have like a reason either to say like plastic and
Oh, actually, that's what I was still reverse DNA replication.
Alright, I like it. Every
yeah,
year.
it's like those strands like when they're like shaped like they're going up and down and like crossing. In one of those strands, that like opens up and like bubbles. It's like an air bubble. It's kind of like bubble wrap. Like you know, like when you get bubble wrap, they go really flat. Like
First year in this house lab, so I'm excited to see how we do
and
it.
then they push out against the
Yeah.
Plans or costumes. The replication level allows
Yeah.
plans or costumes here. I bought a tinsel hat yesterday, which I'm very excited.
Nice.
What's
I love it. Should they wear costumes as well?
This is an unknown.
You gotta keep it like sharp.
I guess my cat. They tolerate it. All right, so DNA at this point, what has been done? What has happened? I'm just gonna stand up here.
We formed a bubble.
It formed a bubble. What do we call that bubble? Yes, the origin of replication has formed the replication bubble. All right, so what does that mean DNA has done? What has happened? Is it a double helix? No, it is unwound. Yes. Right, so DNA has unwound. It has, at the origin of replication, a replication bubble has formed. Right? And so now we have two replication forks, one on either end. And at each replication fork, we are now ready to start replicating our DNA. All right, what proteins have played a role in unwinding our DNA? What's the first one we talked about? DNA helicase, and what does it do? it unwinds the double helix, it breaks those hydrogen bonds, keeping those base pairs together. I think they all come up at once there. All right, we also have single-stranded binding proteins. What do these do? Yes, it keeps the single strands from becoming that double helix again, right? It keeps that area open, all right? And DNA topoisomerase, what is its role here? Prevent supercoiling, yes. All right. All right, so we've talked about how it's unwound. Let's talk about how we're actually going to make another strand of DNA. All right, so at this point, we've talked about this half of the image, right? And I'm intentionally showing you different like artist renditions of how this happens, because I feel like every image depicts these proteins in a different manner. All right, so for example, helicase is now a triangle depicted in this image. But we're going to talk about the rest of replication. What is happening? All right. We have another protein coming into play here, another enzyme. This one's called primase. Right? And the role of primase is to add RNA nucleotides to a certain section of that unwound DNA. Right? So it's adding RNA nucleotides. And that sequence of RNA nucleotides is pretty short. It's about 5 to 10 nucleotides. And we call that section of nucleotides, RNA nucleotides, the primer. All right, so primase is going to come in. It's going to add in a primer. Now what that primer does is it signals to our next enzyme that this is where we need to start making our new strand. Right? It's kind of an area that it's a signal. It tells the next protein where to bind, essentially. Right? Now remember, we have two strands here, right? Because we've unwound a double helix. And the two strands are anti-parallel. They go in different directions. Right? So primase is adding a primer in different directions. Right? We're going to see directionality becoming very important here. So primase can only read 3 to 5. So it builds the complementary primer strand starting at the 5 end, going towards the 3. Right? So that means on one strand, it's going to be building it towards the replication fork. And on the other strand, it's building it away from the replication fork. Right? Because it can only read in a certain direction. It can only read from 3 to 5. All right, so we're adding in these RNA molecules as kind of like a signal saying, this is where we need to start replication. Now eventually those RNA nucleotides will be removed and exchanged with the proper DNA nucleotides. All right, so primase has added in our primer on both sides, on both strands that have been unwound. Our next molecule then, DNA polymerase 3, will then actually sense or know where to start replication by binding to that RNA primer. Right, so the RNA primer tells it where to start. It's then going to read the template strand, which is the parent strand. It was the strand that was originally unwound. And again, it reads in a very specific direction. It reads from 3 to 5, which means it builds our new strand, our daughter strand, starting at the 5' end, working towards the 3' end. So DNA polymerase 3, right, is going to read that template strand. And when it reads a template strand, so for example, maybe it reads in adenine, it's going to know that it needs to put the complementary base pair into place, which would be the thymine. Right? So it knows that thymine needs to go into that position. Maybe the next one it reads is a cytosine, so it needs to place guanine as the complementary nucleotide. right? So the nucleotides, they've already been made, they've already been synthesized, they're around, they're in the vicinity. DNA polymerase will pull in the correct one and start building that new daughter strand according to the instructions on the parent strand, on the template strand. And as it adds in nucleotides, it's going to covalently link them together. And remember, a covalent bond between nucleotides is called a phosphodiester bond. So it's building those phosphodiester bonds, keeping those nucleotides together as it's polymerizing our new molecule. Right? It's building the polymer. It's taking in those monomers and building that polymer. All right, so one of the biggest picture things here is the direction in which it builds. Right? Because again, directionality, super important. It reads 3 to 5, builds 5 to 3. Which means when we build our new strands, right, we have 2 strands running anti-parallel from our parent strands, we're going to build those new strands in different ways, in different directions. All right, so we're going to talk about what's called the leading and the lagging strand. So the leading strand, right, it's the one on the top of this image here. The parent strand at the replication fork is 5 prime. And at the other end, away from the replication fork, is 3 prime. Now DNA polymerase can only read 3 to 5, which means it has to read in the direction away from the replication fork towards the replication fork. Right? So once a primer has been put into place, and it's kind of a darker blue in this image, that primer, the DNA polymerase recognizes that primer, starts building our daughter strand, and it can build that daughter strand in one continuous strand. Right? So as it gets closer to this replication fork, right, the helicase is just going to open up that replication fork even further, and this DNA polymerase can just keep on adding more molecules. Right, so we're creating a single continuous strand on the leading strand. So this is different from the lagging strand. Right, it's different in how many pieces we're building. Right, because DNA is still going to read, or DNA polymerase is still reading in the same manner. Still reading 3 to 5, building 5 to 3. But the lagging strand, which is the opposite, the bottom side of this image on the screen, at the replication fork we have the 3' end of the parent strand. Away from the replication fork we have the 5' end. Well, we can only read 3 to 5. All right, so we might say read, excuse me, my throat is super dry this morning. I'm going to take a drink of water while you write down all of the blanks here. All right, so we have a primer. We'll start with this primer furthest to the right here. DNA polymerase is going to bind to that primer and make a short fragment. Right? At that point, it's going to run into a fragment it's already made. Right? So it's going to leave the lagging strand, rebind further up the road, further towards the replication fork, find another RNA primer, build another segment until it runs into the segment it just previously built. It has to stop. Right? It's going to unbind, find yet another primer, build yet another fragment, right? And so on and so forth. It cannot make a continuous strand. It has to be made in these segments. Right? And these segments are called Okazaki fragments. It's named after the individual that discovered this technique. And these fragments are short comparatively speaking. Right? They're still like 100 to 200 nucleotides long, but compared to the whole length of the DNA molecule, that's relatively short. All right, I know it's hard to visualize when you can't see something moving. So I have a video showing you this happening all at once. All right, I think it'll help put these puzzle pieces together for you here. It does have volume.
DNA is replicated by continuous synthesis of a leading strand and discontinuous synthesis of a lagging strand of the DNA molecule. Coordination between leading and lagging strand synthesis is achieved by the dimerization of DNA polymerase molecules at the replication fork. The DNA polymerase moves with the replication fork. The polymerase at the leading strand template remains attached to the DNA, continuously synthesizing the leading strand. The lagging strand polymerase initiates DNA synthesis at the fork from an RNA primer made by a primase complex. The polymerase elongates the lagging strand in a direction opposite the fork, but stays bound at the fork. As a result, the newly synthesized lagging strand fragment loops out between the polymerase and the fork. Once the polymerase completes an Okazaki fragment, it dissociates from the DNA template. A new primer is produced at the fork. The polymerase reassociates with the template at this position to continue synthesis of the lagging strand. By this mechanism, the two polymerases can add nucleotides to the growing strands at the same time, and at rates up to 1,000 base pairs per second.
All right, so you can see in that video how both the leading and the lagging strand are synthesized at the same time. But now hopefully it makes more sense as to why one of them, the lagging strand, has to build those fragments. All because of the direction at which DNA polymerase reads. All right, we'll review this yet again in a few slides. But let's do a quick review to make sure we understand what's happening. I'll give you a couple of minutes. Bottom 153. Bottom 153.
Oh, they both agree, but the DNA switches is the opposite. Yeah, but the top is 5. And the bottom strand. But it goes, but yes. Question is, can you predict what will go next?
Direction's always opposite in the way that it's explained, right? Like when you,
Yeah.
like in the way it's like, oh, it's going 5 to 3, but realistically it's moving in the direction of 3 to 5. Does that make sense?
Yes.
Like, so yeah, so it's like it's saying, but it's going to be opposite. Like, oh, like 3's here, 5's technically down here.
Yep.
But the leading strand is going towards it, but technically it's 5, 3.
Yes.
Okay.
So it reads in a different direction than it's built.
Right, so it's reading the opposite and moving, they're moving the opposites versus what it's reading versus what it's actually
Yes.
making.
Yep.
And then when you say the lagging, it's reading the opposite, so it's moving your opposite. Or is it still going
It's
the same way?
reading and building in the same direction.
Oh.
It's just that strand, the directionality is opposite of that strand.
Is that why it like went like this way? Is that like when there was this like
Yeah, where it looped out.
right, it's like it's
Yep.
reading this way and going that way?
Nope, it's reading opposite way.
Oh, okay, so it's reading
Reading
and
that
making
way.
this way.
Yep, building in that direction, but building starting at same 5' end, building towards the 3' end.
Okay, so it is still building this way. Or what, it's technically opposite.
It's technically opposite.
It's all just too much.
Yes. Yes.
Leading strand reads from, when I was just a read to lead.
Both leading and lagging strands. The topo DNA polymerase 3 will always read from the 3' end of the parent strand to the 5' end of the parent strand.
How many trick-or-treaters is that? See, these strands not reading.
But that's not that bad.
It is red.
For the first question, I was saying 5 to 3.
So you can see it's starting here at the 3' end of this.
Yeah, it's like in, when
It's
they talk
starting
about
at
it,
the
they
3'
say
end of the lane.
from 3 to 5, so it reads the
But because the
opposite.
parent strands are
So it's like,
anti-parallel.
oh, yes, it's replicating 3 to 5, so it's reading, or it's moving 5 to 3, but reading 3 to 5. It's all opposite.
And is there 2 numbers just like not that? Yeah, so which way is it the new strand?
I would say
Replicated 5.
The best way to, because it's just, I'm watching the videos on YouTube.
Okay,
Find
so
different
you
ways
use
to better
the
illustrate.
lagging.
And I didn't do ChatGPT the whole time. I said symbolize it, what am I looking at? We're like, yes.
5 to 3, but it's
Yes, it's all about directionality. You have to understand directionality to understand
it's building 5 to 3 and reading 5.
I've got a bike, but unless you expect me to bike.
Remember all the opposites.
No, this is no, but I have a basket.
All right, so we're going to review questions.
Like when we were going up, we were
What
up.
direction is the new strand of DNA replication? I want to rephrase this. What direction is the new strand of DNA being built?
It's so, I don't understand it.
Yes, it builds the 5' end first, and then adds towards the 3' end. Right? Even though it reads the 3' end, reading towards the 5' end. Right? So I keep saying directionality is important. This is why it's important.
Confused.
All right. So then, because of that directionality, how is replication different in the leading versus the lagging strand?
Wait, what's the first answer?
Which one has the Okazaki fragments? The lagging strand. So the lagging strand has to make fragments, those Okazaki fragments. So that means that the leading strand is just going to build one continuously new strand of DNA. All right. Directionality, important. I would say if this is confusing, I think the best way to study this is just to watch videos of it happening in real time, right? Pictures are great, but they're just a snapshot in time. They don't show it actually working. Look up some YouTube videos. There's great illustrations out there. Maybe somebody might explain it in a way that clicks with your mind a little bit better, right? But visualizations are super important when studying here. All right, so at this point, right, we have a DNA, some replicated DNA, that also has some RNA nucleotides thrown in, right? Because remember, we started at that primer, and that primer is made out of RNA. So what can you predict is going to happen next? Yeah, we have to get rid of those RNAs. We don't want our RNA nucleotides in our replicated DNA. We have to replace it with DNA. Right, which is what?
Oh, I should have took a picture.
There we go. I forgot I had the answers up here. I'll leave them up here for a few seconds here. All right, so leading strand makes one continuous, lagging makes fragments because of the directionalities that DNA polymerase reads and builds. All right. So we have our segments of replicated DNA made. Those segments still include the primer sequence, which has RNA nucleotides. We need to get rid of those RNA nucleotides. We have yet another polymerase. This one's DNA polymerase 1.
The first one was
The first one was 3, yes. Again, they are numbered not consistent with the order in which they play a role. Right, so DNA polymerase 1, right, after we've replicated our DNA, we have a daughter strand, is going to come in and recognize that RNA primer. Right, so on the bottom image, that RNA primer is highlighted in green here. Right, and that primer sequence is inside of our DNA polymerase 1 there. Right. It's going to get rid of, with the help of some other enzymes, get rid of the RNA nucleotides and replace it with the appropriate DNA nucleotides. Right? So again, it's using the parent strand as the template. Right? So if the parent strand that originally was bound to RNA had cytosine on it, right, DNA polymerase 1 knows that it has to add in 1. We're still following that complementary base pairing rule. Right, so we've now swapped out the RNA nucleotides for the DNA nucleotides. So now we have a segment of DNA nucleotides that used to be where the primer is at. That segment is hydrogenally bound to the parent strand. Right, so it's like the rungs of the ladder have been made. The sides of the ladder have also been made, so the nucleotides within that segment have also been covalently bound together. We've made those phosphodiester bonds. But what RNA polymerase 1 cannot do is make the bond, the covalent bond, from the nucleotides that it just replaced, and it cannot bind it to the nucleotides that DNA polymerase 3 added. So there's going to be a gap, right? And we have to fix that gap. We do not want a gap, which is where our next enzyme plays a role, ligase. All right, so ligase is going to come in, and it's going to fix that gap between where the primer used to be, that is now a DNA sequence, and between that sequence that RNA polymerase 3 had already made. Right? We kind of think of ligase as like the glue. It glues it together. So that after ligase has come through, right, we now have a continuous strand on both sides. We have glued all of our fragments together. We no longer have any Okazaki fragments. We no longer have any gaps between primers where the primers used to be in our DNA sequence. Everything is glued together. All right. I think ligase is, in my mind, ligase is one of the easier ones to remember because it's a short name. It doesn't have a complicated name like topoisomerase. And it does a very simple job. It just glues the ends together. All right. I get another video because, like I said, visualization I think is important in helping you learn this concept. This is going to go over the entire process of DNA replication.
DNA is a molecule made up of two strands, twisted around each other in a double helix shape. Each strand is made up of a sequence of 4 chemical bases, represented by the letters A, C, G, and T. The two strands are complementary. This means that wherever there's a T in one strand, there will be an A in the opposite strand, and wherever there's a C, there will be a G in the other strand. Each strand has a 5' end and a 3' end. The two strands run in opposite directions. This determines how each strand of DNA is replicated. The first step in DNA replication is to separate the two strands. This unzipping is done by an enzyme called helicase and results in the formation of a replication fork. The separated strands each provide a template for creating a new strand of DNA. An enzyme called primase starts the process. This enzyme makes a small piece of RNA called a primer. This marks the starting point for the construction of the new strand of DNA. An enzyme called DNA polymerase binds to the primer and will make the new strand of DNA. DNA polymerase can only add DNA bases in one direction, from the 5' end to the 3' end. One of the new strands of DNA, the leading strand, is made continuously, the DNA polymerase adding bases one by one in the 5' to 3' direction. The other strand, the lagging strand, cannot be made in this continuous way because it runs in the opposite direction. The DNA polymerase can therefore only make this strand in a series of small chunks called Okazaki fragments. Each fragment is started with an RNA primer. DNA polymerase then adds a short row of DNA bases in the 5' to 3' direction. The next primer is then added further down the lagging strand. Another Okazaki fragment is then made, and the process is repeated again. Once the new DNA has been made, the enzyme exonuclease removes all the RNA primers from both strands of DNA. Another DNA polymerase enzyme then fills in the gaps that are left behind with DNA. Finally, the enzyme DNA ligase seals up the fragments of DNA in both strands to form a continuous double strand. DNA replication is described as semi-conservative because each DNA molecule is made up of one old, conserved strand of DNA and one new one.
All right, so that hopefully puts everything together for you. Now, it did talk about exonuclease. We didn't talk about it in the slides. I wanted to expect you to know it, where it actually, that's what removes the RNA primers. I want to end our talk about DNA replication by saying DNA replication is very accurate, right? There are methods by which we make sure that we are adding in the right nucleotide. The first method is simply that hydrogen bonding we talked about between base pairs, right? A and T can only make 2 hydrogen bonds. G and C can make 3, right? So if we have a base that can make 2, trying to pair up with a base that makes 3, right? That connections are just not going to be as great. It's not going to be as stable, right? So that in and of itself is a good method for error checking. It's not going to be put together correctly, right? But DNA polymerase actually has a mechanism of being able to proofread, right? So first of all, it's very unlikely to form pairs if they are mismatched. Right? DNA polymerase knows what it's doing. It knows how to read the template strand. So it's very unlikely to make a mistake. But if it does make a mistake, it has the ability to proofread. It can actually back up, digest the incorrect nucleotide, and put the correct nucleotide into place. Right? Now, there are other repair mechanisms as well. There's other DNA repair enzymes. Right, but these are some of the 3 of the most broad categories as to why replication is very accurate. All right, so let's do our last review on DNA replication. I shouldn't say our last review. Our content check is also a review. But I want you to tell me what proteins are involved in building this strand.
The other one. And then the other one. Topoisomerase. And then I don't know the fourth.
So is topoisomerase involved in building the strand?
I didn't realize we were doing so There's no 2. We discovered this one first. We discovered this one. There's no second. I don't know how this one goes there, but we don't know the other one. There probably is a second one. It's just not I really like when scientists is like, I discovered this thing first. I'm gonna call it this, and then it doesn't matter what order it is. I'm going to know the first. Are they found first? Yes, they're all proteins. Instead of just being like, we should not write this or because they think it'll confuse the group. Why would this be like,
Perimeter is 1, or just perimeter. It doesn't need a number. Does it really need a number?
well, because there's 2 of them.
Oh, we don't really discover this.
Right, but they add the one later. They add the one later for like order, but I feel like they should order it based off of. It starts off from. And I really don't think it's hard scientists who just don't use this and they'll get confused. But like, you guys have been dying for 30 years. Let's think about the future. Let's think about the youth. You're gonna get used to that. I promise you it's not.
like the sound. They all ended up not fine, and then yeah, it's really different. It's like well, I was should be if I was a scientist, I would absolutely be like, I am the best kind of scientist. And I think everyone else, every other kind of scientist is that's what I was thinking. Scientists.
Alright. So this question is asking proteins used in building the new strand. Right, so we're going to ignore all those proteins used in unwinding the parent strand. What's one of those proteins used in building the strand? What protein do we need to set the start point? We need primase. Primase to make the RNA primer, yes. I know when I take this, all the answers are, I think, going to come up at once here. So what's another protein that is involved in building the strand? Yeah, DNA polymerase 3. What does DNA polymerase 3 do? Yeah, it builds it. It does the bulk of the work, right? It adds in all of our nucleotides, most of our nucleotides. Okay. So then DNA polymerase 1, what is the role here? Yeah, it replaces where the RNA nucleotides were, it replaces it with the DNA nucleotides. Yep. All right, so now everything's DNA. And then the ligase comes along, and what's the role of ligase? Puts together all the fragments, glues all the fragments together. Yes, so now there are no gaps. It is one continuous line of newly built DNA. All right. We have a content check due today. And so I've allotted 15 minutes, I think my last class 20 minutes. So I'll give you 20 minutes to work on this here. So you can work on the in portion, the part 2, as well as complete the online one if you haven't yet completed it. You should have all the information you need now. After 20 minutes, if you need to leave and come back, that's fine. We'll do a quick more slide with content. I mean, you don't have to. That's 2 points.
Is this sugar in DNA? That's not that's not
It's a pentose sugar.
a pentose,
It's a
right?
5-carbon sugar.
Pentose is 5.
Yeah, yeah.
And then it's not, oh gosh, that's what it would be. Already. Okay, yeah, thank you.
I sure want to know those. Okay. Is it China? Okay.
So we talked about
Structured DNA replication.
I should say, okay, during cell
And then, wait, this is just you can
yes. It wouldn't let me do this thing twice, so I had to.
This is a little.
maybe it's not pencil. I just need to make this big. I was thinking we were doing steps.
No.
Draw DNA replication. All right. Do you like that? I think it's good. No, I'm just thinking. Oh, yeah. What do we draw? That looks good. Yeah. That was like only a little bit. And like, how do you choose that?
You're struggling to start. I'm going to draw the start up on the board.
Yes, start it. Please. Start it now. Okay, we got it. Okay, I'm gonna start that.
But maybe a good way, you know? Like, he's like this Southern Baptist comedian.
That doesn't sound
Okay, when I say it like that. But he did okay. Okay.
Yeah.
What's his name? Casey. He's very passionate about what he does. And he's also a very gracious person.
Yeah, let's make this one. I only took it because I thought I had helped. What's your Oh my god, is it like ancient? Oh, okay, it's ancient. Go only ancient Greek.
Yes, that's like.
We've discussed this.
Like that's the bubble.
As in class. Oh, and?
Well, we don't have the money.
So you drew the whole thing versus just like a half where like we normally
Yeah,
draw a
funding
half.
hasn't come through
Yep.
yet.
No.
Because there's a point I want to make after we're done with this.
Okay, okay.
Yeah, this is, so you need to get going.
Okay, so it's almost like we have only.
Wait, so it's only
Oh, wait,
one
I'm trying to
inch?
this way.
You do?
Yeah, I think she's gonna.
Okay. What I draw, hold on.
Why am I signed out? It's always 5
Depends. Well, it reads that.
That can't be right.
You should draw, yes.
I will draw like the reading strand.
And it might help you out like you look at it or just everything else with that color. You don't have to primates, but I'm gonna let you work through it a little bit here.
Got me this one. This is because we're used to only seeing half of it. No, because these are going opposite ways. It's unzipping like this. Okay. I did it.
I just did it the opposite way. I did it more the way because like it's just it makes more sense because that's what we've been doing this whole time. This slide on the top, three at the bottom. So I think I'm going left to right. That makes sense.
Because this is the bubble. Yeah, but that's when it's going this way. So if it's going this way, it's going to be opposite. Because it splits the bubble in the middle, and then they go out. They're doing their own thing.
Yeah.
Okay. And you just look at that like that. So this is like this. So when it's going two opposite ways, we have to help both of the stay. Yeah. But it's building from each other.
Where is it? That is.
So like this isn't going.
Don't know, you don't need to drive. Oh, wait,
It's
yeah.
starting at which one?
Whoever is 3.
Yeah, but you don't need to know where is one.
Which?
I do.
Whoever is 3.
That's the one that goes 3 to 5. Or 5 to
What is it?
sphere.
That? So it's the minus.
They're all kind of spheres if you think about it. They're just kind of like. They're really all donuts. It doesn't matter how you present them. You just have to label them.
Oh, here it is. Oh, so that, it's a big chunker.
Where? Dude, just I just drew it as a donut.
Does it matter which side?
It's on the second one. It just depends
Oh,
which way you
fine.
It's on both of them.
It's on both.
Oh,
So it just works better, okay, yeah.
So when I drew it up there, I drew like this dash.
There's one of each end.
Okay, that's like the ultimate start point. That's where it like
Because look,
opened
it's
up.
on the.
Okay, and it's going outwards from there.
Yeah, the color right.
Oh, so it goes here.
So
and
the
here,
leading
and here?
strand.
No, there's two. You only have part of it. The bubble.
Because this one is in the opposite direction.
You don't have to.
Leading strand would also go
If
here.
you can just draw one on each side, because it does it.
Yeah.
It's putting it on an
Yeah.
experiment.
Right. But this is the thing with the front.
So you have
Or is it?
yeah.
I didn't specify. Okay, I drew it up on the board because I want to make a point.
And this is the starting spot, and it's going from both ways, so it goes away from each other. So when you then have that, The leading one is the one that when you look at the other side like this, because it's going continuous.
It is manual.
From where it's unzipping, because it's unzipping this way. So it's able to continuously go 35 because it reads 35. But on this side, the top way is like 5 to 3. So that's not the leading one on the other side. Because you still only read 3 to 5.
Yeah.
Because I have some opposite side, it's on the bottom.
So they're just opposite.
Yeah. Because they're going outwards.
Are you
See, this one.
typically I do it when I'm home?
Polymerase.
Wait, this one? Yeah. Nice. Yes. When I'm home for I will put it up before Halloween, but not this year.
Like one for each
Yes,
side.
if you have the drawing done, or when you get it done, make a pile up in front. You have to speak on it if you're going.
I would draw the arrow, which would go in straight. But other than that.
So it's after primer. We're gonna build
Oh, I guess I should have a little loop.
off the primer and the reeds or a bunch of them. The primer's always gonna be like 5. What is it? Different on either side?
If it does all
That was all right.
yeah. I'll leave the test so you put extra.
My color goes down.
Exactly.
And then I painted it?
I need to write it.
Don't have markers.
I do, I just don't care.
Let me go up to
No. Only class I cared enough to color code is my EMT one. I mean,
So, it's slash when you use are you gonna be excited?
I'm dad. I'm just gonna look at this side.
that's not doctor.
Teenage polymerase.
That's paramedics.
I don't read the clear.
Typically not because they have the actual materials.
Yeah, but you don't have that.
An ER doctor is not gonna have an ER doctor is not gonna have any stuff for them. My grandma was an ER doctor for 20 years. I guarantee you he could not. No, I forgot them apart that. My friend's in school to become a paramedic because he got his EMT license.
This side
He
is
works at a
the
hospital
primer.
now.
The 3-5, it can still only move this way.
Everything's so crumpled
But
because
it's not moving towards me.
No, because you can't own a private ambulance. You can if it's like an abandoned range, but most people don't.
Opposite direction.
Or like live with their
Now
parents.
this thing at the top
Or wives. They also all have this. It was like the little.
Now it's reading for 5.
The you know the firefighter like that? And it's also an EMT like that. Because every EMT wants to be a firefighter because they get paid more. Okay. It hasn't been said yet. But to the side, go paramedic training. And he always get paid like.
So to like marinate in that information.
Like 9 months.
I spent a year doing it, but it was like a great hospital.
So if you were just.
Yeah. It is extremely hard.
I mean, yeah.
I was also in a very funded program, so
the only one that's on the leading is the SSB program.
like we had a fake ambulance in our class with DNA redo.
All it is
Yeah.
otherwise, this is all the stuff that needs the new one.
Not the lights.
Otherwise, this one's bleeding. It's not.
We also had other things. When I went on my ride-alongs with actual EMTs, they assumed I had the register before. Yeah, I touched on the daily.
So you're like,
I've been in it.
No.
Someone drew my blood, but that wasn't one of my classmates. Don't draw blood. That was one of the.
Yeah, I mean, I've did some 2 different diagrams, so
I can't do it, so I got help with my second I looked it up and I took it, but it's supposed to
Where is it?
I didn't get blood before because I was like, I don't know if I'm gonna be, there's like a slight chance I can transfer it my blood. And I was like, I would rather not than ever do that to anyone. And then once I figured out what it was, I looked it up and they're like, no, you could pass out. And I was like, oh. Which is a really bad. I could pass out not doing So when the Bobby students were just they were doing it probably better than all of the nurses I've had take my blood. And they were talking about this like
Yeah, I would be fine for a long.
because they were just testing how to do the IVs and then how to do finger codes. Which they couldn't for me because I'm like a little piece of shit. So, you know, simple, easy, students pass out. Which is like my gotcha. I'd come back like both arms, like patting on them, and I'd immediately get back into my EMT stuff. I'd be like, I've got a patient to deal with, bye. Or is it one? Do you have one? My siblings also do that. They're in second year. The fragments are in the lag. They're taking over your job.
I did like my language strand with as fragments.
They're doing it to get money so that they can. It's called
My face is
Wait, I probably got that. They're at St. Louis. Our rival. I don't know, but our other rival, Carlson. Oh. Who does? And my friend goes there, and I was like, oh, yeah. We beat their asses. That makes sense. Paved the way in what?
We still have 5 minutes. I know the timer's up, but we still have 5
They
minutes.
both work in the medical field though. And my friend actually.
Yeah, no, I'm not. I got
But
nothing going
technically
on.
I'm a medical professional, so who's really good at it?
I mean,
Exactly. I just got my
I mean,
degree.
hell, if I drew an essay.
So technically I have a college degree.
yours is a lot cleaner than ours. My entire paper's
My friend got one. She has a degree. She's actually in college, so. Both of us have degrees before college. Yeah, well, I mean, she was already working which is cool. But then also, that's why I decided to go to college instead of being a paramedic. I was like,
Yes. I believe once I take time
I'm
to interpret it, I will
think
be.
I'm-
I just
I
was
think
like,
I'm-
I need transition. I need to transition because I will genuinely go batshit crazy.
And I can't learn this, I can't learn this on the whiteboard.
I never went. I did not want to. I'm not making it.
But she gets light and dark.
I'm putting to death for it. Which is presumably like my mom thinks I'm my dad's like, oh, you should stop. So it's not good. Guys. On the other side, if I do it that
Fine, we will resume content at 11.
the tank.
Later.
Dude, imagine you're like struggling.
In my back.
The second I get it, I'm raw. And then a paraglider shooting up for 5 seconds later, and I fly off into the distance. I just don't want to be in a wheelchair. Well, see, I mean, I'm a runner. Yeah, no, it's actually the only thing that's kept me from getting worse.
It won't matter.
But the thing is, if I was in a wheelchair, I'd have to sit all day. That's not how it works.
At least on the bright side now, they have so many costume options for people in wheelchairs.
You're so right.
Well, compared to like the last 10 years.
I'm waiting with blue guys. That'd be ka-chow. Ka-chow, they say, I run over a small child in my wheelchair.
Yes. But I still got
I feel like he's just saying, I just don't want to stand again. If you put me in a wheelchair, I'm just not. I'm not even gonna try. I was wheeled around in a wheelchair once. It was on my way to get an I've had 3. It is. They don't play any music. So the MRI is like, imagine. No. I have that. So, the best way I can describe it.
Okay, thank you, Raina. Thank you.
To be fair, mine was on my knee, so I did it. Mine was on my back every single time. They did 3. It's like being in a coffin, but like smaller. It's like, and it's like this far away from your face. They put a rag on my eyes and then I don't look, and then I got it and the rag immediately slipped on my, and I was like, you have a little like button in your hand that you can press to tell them to stop. And then it's like, you have earphones in, but there's like, it sounds like there's 6 speakers around you full on with like 1 second of a note step song on repeat. And they keep switching what part of the song it is. That's what the experience was. Deoxyribose. Deoxyribose is like, and then RNA is here. Yeah. Yeah. You know Deoxys from Pokemon? So ribose is that, but without the deoxy. It's got DNA.
What generation is that?
I don't know.
I played Pokemon Go and I like the first 2 or 3 before I stopped playing it, and they released more apps.
I played Pokemon Go like occasionally for a year, and then my cousin would not shut up about it, and I deleted it.
My husband's big into it still. I'm just giving something to do when we want.
This is Deoxys.
All right, let's continue on with the lecture here. We don't have much left. I wanted to talk about this content a little bit. So I have drawn an example up on the board. Because I want you to recognize that many of you probably just drew one side or this happening at one replication fork. And that is fine. You'll still get full credit. But I want you to understand it's happening on both sides. Right? And so on both sides, both replication forks, right, there's going to be a leading and a lagging strand, but they're actually flipped because of directionality. Right? So if we think about our leading strand, right, it's made continuously. So on the top of this drawing is where I draw my leading strand, right? It's built from the 5' to the 3', and it's going to continue building as DNA helicase unzips more DNA, makes more DNA available, right? But our lagging strand, right, still built 5 to 3, but because of the anti-parallel nature of directionality here, right, it has to build in the opposite direction, thus building the segments. So if we look at the other side, the other replication fork, the leading strand would actually be on the bottom of what I drew here, right? Because again, it can only read 3 to 5, build 5 to 3. So it's still building in the 5 to 3 direction, right? And then the Okazaki fragments, if I were to draw them, which I didn't for that second half, would be at the top, right? And still be built in the same direction, right? 5 to 3, but that direction would be opposite because of the anti-parallel nature of the parent strands, right? So this is happening all at once, right? There are 2 replication forks, and it's kind of like it's swapped on either side because of the directionality. And you might have to look at this a little bit longer and really like walk yourself through it. I know I have to walk myself through directionality every time, like, okay, reads 5 to 3, or builds 5 to 3, reads 3 to 5, which is my 3, and which is my 5, and now I gotta flip it, right? It might take a little while, that's okay. But I want you to understand, it's happening on both sides. Are there any questions about DNA replication? All right, well, we're going to look at kind of the other side of DNA replication. Why did DNA replication occur? Okay. Well, DNA replication occurs because cells divide, right? Cells can divide into new cells, which means we have to replicate our DNA so that our new cells also have a set of DNA. All right, so we're going to start talking about the cell cycle. We won't actually get to the cell cycle today, but we're going to introduce some concepts, right, about chromosomes, still thinking about DNA replication, that will apply when we talk about mitosis and meiosis. All right, so we're still looking at chromosomes. We're still looking at eukaryotic chromosomes. Right? Chromosomes play a huge role in cell division. We know chromosomes, they are compact, but there are times when they are more compact than other times. Right? When a cell gets ready to divide, these chromosomes become super compact. And we can see that in this image here. Right? This cell right in the middle, this cell is not dividing. Right? So these chromosomes, right, are euchromatin. Right? They're compact, but not super compact. But the cells that are actively dividing, right, on either side of that middle cell, we can start to see very distinct chromosomes now because they become super compact, because they are now heterochromatic. Right? There's actually a field of biology that if you were looking at chromosomes, you could get into cytogenetics, which is looking at chromosomes under the microscope. Right? Just kind of like we're doing with this microscope slide image I have up on the screen. All right, so looking at chromosomes still, when they're getting ready to divide, they become extra compact. there's still some more terminology that we have to talk about chromosomes. I know we've learned so much already, but there's yet more. All right, we've talked about homologous pairs, right? So one chromosome comes from mom, one comes from dad, contains the same type of genetic information. They are a pair. Okay? We can assign different ploidy levels to our types of chromosomes and how many types are in a grouping. All right, so if we have a pair, if we have a homologous pair, that means we have two chromosomes with the same type of information, but only two. So if we have two chromosomes, right, that cell would be diploid. So for each set of chromosomes, right, there are two chromosomes for each type of information. Again, one from mom, one from dad. Right, so those would be diploid cells. Now when a cell goes through meiosis, right, or if there's a mistake that happens during mitosis or meiosis, we could end up with a cell that is haploid, meaning it only has one chromosome per type of genetic information. Right, so now you don't have one from mom, one from dad, you just have one, and it's either from your mom or from your dad. Right, it's kind of random on which is which. Right, but you only have one chromosome representing that type of information. That would be a haploid cell. Now there's other levels of polarity, right? A cell doesn't have to be haploid or diploid, right? There are cells out there that could be triploid, right? In humans, this is very rare to have cells that are triploid. But triploid meaning that for each set of chromosomes, you actually have 3 chromosomes in each set, right? You have 3. Again, this is very rare for an entire DNA sequence to be triploid in humans. A lot of times in humans, if we see triploidy, it's only going to be in one set. Like one set out of our 23 sets will be triploid. But there are organisms out there that are triploid organisms. Right? All of the chromosomes have 3 sets. There's organisms out there that are tetraploids. Right? So they have 4 sets for each of them. Right? There are organisms out there that have like 17 as their ploidy level. I don't even know what the terminology is. I would write it shorthand as 17N. Right? Meaning for each set they have 17 chromosomes that contain that type of information. Right? Having that large number is more common in plants than in mammals. Right? So there's a lot of variation. Now when we're talking about mitosis and meiosis, we're going to stick to haploid and diploid, and we'll briefly talk about triploidy in humans when we talk about mistakes in mitosis and meiosis. All right. Humans, and we're going to stick with humans now. Humans do have 23 pairs of chromosomes. Okay? 22 of these pairs we call autosomes. Right? So 22 of these contain most of our genetic information. Right? Height, hair color, eye color, all the types of genetic information that's going to be transferred from your parents. But we have one pair which is our sex chromosomes. Right? So this is where it, when you're born, it determines if you are male or female. Right? Either your XX or your XY. Right? So we have one set that determines our sex-specific genes, male versus female. Right? All the others we would call our autosomes. Now if you're a scientist, maybe you're a scientist in that cytogenetics, so you're looking at chromosomes under microscopes. Maybe you're trying to determine the ploidy level for an organism, or how many chromosomes they have. Right? You're going to develop what's known as a karyotype. And I actually have 2 images of karyotypes up here. Right? A karyotype is just a visual depiction of every single chromosome, right? How many are in each pair or in each grouping, right? And their size. So if we look at the human karyotype, which is the one up top, we can see that they're ordered by size, Our first set of chromosomes, or homologous pair here, are very long, whereas our last set tend to be really short, our 21 and 22. Okay. Other organisms that have different numbers of sets of chromosomes, like this one down here, this is coming from an organism that actually has 40 sets of chromosomes, so it's not a human. Right? But again, it's numbered, and it's ordered from largest to smallest. Right? So if you see an image like this would be called its karyotype. This gives us more information on how many, are they haploid, diploid, triploid, and what are their relative sizes. All right, let's do some practice, and I know I've already asked you to label chromosomes, but we're going to do it again, and I'll give you a couple minutes here.
And then that's a diploid and a triploid. Oh, yes. What are the other chromosomes? Just because they're
Is the product of a moose and a donkey?
a horse and a donkey. And so a horse has 2 more chromosomes than a donkey. But it's so solid.
Oh, it just says.
So what would moose
Okay. Yeah, sure.
So to breed an animal to have a certain amount of chromosomes. And I doubt that is the answer. But if it did, it would probably a monkey exists. Is that what you want? I don't know. She briefly talked about our cat.
Making sense.
Dad. I want dad. Oh, my sister sent me her dad.
She started her first ever real job, where she works on Monday. She's the head of marketing, because she's the only one in the marketing department. We're very proud. She's very excited. And she is supposedly going on her first work trip.
My dad.
All right, let's review this slide here. So if I were to ask you what is A, what would you tell me? Most of us would have the answer. All right, let's skip A. We'll come back to A. Let's do B. Centromeres. All right, we all have B. What about C? Sister chromatids, right, either side of one of those structures. And then D would be a homologous pair, yes. Right? We have a set that contains the same type of genetic information, just one from mom, one from dad, right? Different variations on it. So what would A be? A would just be a chromosome. Yeah, it's one chromosome out of the 2 that are up there. Yes. All right, I have even at the bottom about ploidy levels. So, there we go. Ploidy level for this first set here. What would you say the ploidy level is? It is diploid. Yes, for each homologous pair, there's only a pair. All right? So that one would be diploid. What about the next one? Triploid. And if you don't remember the name, you can just do the number. So 3N. So in this case, we don't have a homologous pair. We have a homologous set. And that set is of 3 each. All right. Any questions? All right. We're going to keep practicing these terms, especially next week. I'm actually going to stop there because that's where my last class stopped. But I do want to remind you of what's due. Right, exam corrections, scientific literacy due. I'll send out that email with updated instructions. And that modified class schedule. So we actually meet 9:30 to 10:50 next Tuesday. So it's a little short next week.